This video is for class 10th genetics. As we all know, we have started the chapter genetics to learn. Today, we are entering to the technical part of the genetics. We have seen about the basic terms, we have seen about the technical terms of the genetics. Now, we will see the crosses of genetics. So, first cross is the monohybrid cross. I have written here monohybrid cross. Monohybrid cross, as earlier it was informed to you, monohybrid cross means the cross between two parents with single character under consideration. Means cross will be there with two parents, between two parents, but only one character will be taken under consideration. How? Here it is taken tall and dwarf, capital T and small t. Single parents taken. Two parents taken with single character. Means this is one individual parent, single parent. This is one individual parent, single parent. So two parents are taken with single character under consideration. Now what happened? What we did here is capital T cross small t. So here the cross will be taking place, and see how I have made the Punnett square. Punnett square is the, I have already informed that Punnett square is the matrix or the network of rows and columns where we are sharing the number of progenies possible. Number of progenies possible is shared under this Punnett square. So what happened here is, see, capital T one side, small t one side. These two parents are the pure parents, pure line. They are following pure line genetics, or they are following pure formation. Why? Because this is also capital T, this is also small t. We cannot find anything like this over here. We cannot find. Until and unless cross is not taking place, we cannot find anything like this. Capital T, small t, we cannot find. Both the parents are following pure line genetics. So that is why they are homozygous. Means capital T is homozygous tall, and small t is homozygous dwarf. Dwarf means short heighted. So what happens? Capital T cross small t. Now what we did? We made the Punnett square, capital T, small t. And for your consideration, again I am making the cross. You please see to it, so that you should not make any kind of mistakes, or else you should not have any kind of difficulty. So what I am doing here is, for the F1 generation, what is the definition of F1 generation? F1 generation means cross between two pure line parents. So capital T, small t. Just multiply. Capital T, small t, again capital T, small t. Just multiply. Understood? So this is for F1 generation we have found. What kind of progenies or offsprings we have found? What kind of young ones or progenies or offsprings we have found in the F1 generation? We have found capital T, small t, capital T, small t. All are equal. Means since capital T is dominant over small t, so which characters will be shown? Which character will be shown? Long means tall or dwarf? Of course, tall character will be shown. Dwarf won't be shown. Why? Because capital T is dominant. This capital T was dominant, and this small t was recessive. So if two genes are present and one is dominant and another one is recessive, we all know very nicely only the dominant gene will be expressed. So here which gene is expressed? Capital T, small t. Of course capital T will express, and small t will be recessive. It will be suppressed. Capital T will express, small t will be suppressed. Now what happens? This progeny as formed, it is known as heterozygous tall. Why heterozygous tall? What is heterozygous? What I have explained in the earlier video. In the heterozygous means the genes are not made up of, means genes are not having the same genotype. Means not capital T or not small t. They will be mixed. Capital T, small t. Means when the genes are not having similar kind of genotypes, or it is not having similar pair of characters. It is having dissimilar pair of characters, and out of this dissimilar pair, the one which is dominant will express, the one which is recessive won't be expressed. I guess it is understood in the F1 generation what I did. Two parents I have taken, and one is at the top, another one is at the left side, and then just multiplied. Capital T small t, capital T small t, capital T small t, capital T small t. Next, this is how the F1 generation ends. Next, F2 generation. In F2 generation, what I did? What is the definition of F2 generation? F2 generation simply means the cross between the progenies as obtained from F1 generation. Cross between the progenies as obtained from F1 generation. So from F1 generation we obtained 4 progenies, capital T small t, capital T small t, 4 progenies. Cross between them. Any two you select, either this one or this one, or this one or this one, any two you select, any two you can select. Or through this one or this one or this one, randomly any two. Now, capital T small t, capital T small t. For your explanation, again I shall rub it and I shall show you. So what happens here? Just a multiplication again. So capital T, capital T, capital T small t, capital T small t, small t, small t. Now, this is completely different from this one. In the F2 generation, we have crossed the progenies. So what happened here? Capital T, capital T, capital T small t, capital T small t, small t, small t. It means here what happened? The progenies as formed, there will be 3 tall and 1 dwarf phenotype. Now we will be talking about the ratio, phenotypic ratio and genotypic ratio. Phenotypic ratio means physically how the plants are looking. So physically this one is also tall, homozygous tall because sharing same genotype. Heterozygous tall, sharing different genes. Heterozygous tall, sharing different genes. But all 3 are tall. We cannot say the 3 are dwarf. Physically, phenotypically, we can feel that 3 tall plants are there. So 3 is to 1. Why? Because only 1 dwarf. Because we know very nicely small t was dwarf. So out of the 4 boxes, 3 are tall, 1 is dwarf. I hope you can understand this. What I have said, capital T is tall, homozygous tall. Capital T small t, heterozygous tall because the genes are different. Capital T small t, again the genes are different, heterozygous tall. So 3 is to 1. Now talking about genotypic ratio. How this genotypic ratio happens? Now we need to see genotypically. So how many capital T is there? 1. Because it is homozygous. How many capital T small t is there? 2. How many small t is there? 1. So 1 is to 2 is to 1. For all the monohybrid types of crosses, only this ratio is possible. Other than that, no ratio will be there. Especially in the F2 generation. I guess it's clear for everyone. I hope it's clear. So again, as a summarizing, I am telling capital T, small t, 2 pure line parents crossed capital T small t, capital T small t, capital T small t. F1 generation complete because cross between 2 parents. Now in the F2 generation, cross between the progenies, capital T, capital T small t, capital T small t. Cross between the progenies as obtained from F1 generation. From F1 generation, we have obtained how many progenies total? 4. Any randomly 2 we will pick, and then there will be a cross. So capital T, homozygous tall, capital T small t, heterozygous tall, capital T small t, heterozygous tall, small t, homozygous dwarf. Physically, if at all we will see, we can see 3 tall. So phenotypic means physically. So phenotypic means physically, whatever we are seeing from outside, so we can find 3 talls. So 3, 1 dwarf, 1. Genotypic ratio means how is the genotype, whether it is homozygous or heterozygous, that one also we have to see in genotypic ratio. So how many capital T? 1. How many capital T small t? 2. How many small t? 1. I guess it is understood. In this monohybrid cross only, Mendel has explained the law of segregation also. Law of segregation means, please listen to it very carefully. Law of segregation means the gametes, the genes especially, we will see the genes first, later on the gametes. The genes stays together according to law of segregation. Segregation means separation. So listen to it carefully. According to law of segregation, the genes stays together, but at the time of formation of gametes, the genes will separate. The gene will stay together, but when the gamete will start to form, the genes will be separated. So here, very nicely you can see whatever I'm saying here. Capital T was one parent, small t was one parent. When the gametes has been separated for these two parents, when the gamete has been separated, what happened? One parent, the capital T got capital T as a gamete. Small t parent got small t only as a gamete, because they were following pure line. There was no mixing up of the genes. So law of segregation is also called as law of purity of gametes. Law of purity of gametes means the genes stays together, but at the time of formation of gametes, the gene separates. Means there is no chance of hybridization of the genes. Genes will not hybridized. There is no chance of hybridization. There is no chance of mixing up of the genes. That is why it is called as law of purity of gametes or law of segregation. Why? Because these two were different parents, but at the time of formation of gametes, capital T was having only capital T as their gamete, and small t was having only small t as their gamete. So what happened? What we can conclude from this? That when the time of formation of gametes comes for these two parents, automatically the gametes were separated. When the time of formation of gametes came, the genes separated. Before that, the genes were together. But at the time of formation of the gametes, the genes separated. At the time of formation of gametes only. Before that, the genes was together. So one got capital T, one got small t. In the F1 generation, it was found. But what happened in F2 generation? How can we prove the same law over here? Because we got one small t also. Why? Because at the time when these progenies were crossed, and when their gametes were produced, that time what happened? Let's say this one was one parent and cross take place between this. Two parents were there. And when the time of formation of gametes came, one parent got T also, and one parent got capital T also, another parent got capital T as well as small t also. So what happened? We got one dwarf. But here in F1 generation, we haven't seen any dwarf. All were heterozygous tall. But here we can see why. Because at that time of formation, what happened? Capital T was also shared as a gene, as well as small t was also separated as a gene to a particular gamete. That's the reason why we got tall also, we got dwarf also in the F2 generation. I hope this video is clear. Next video will be uploaded very soon. Kindly understand the video, match it with your text. Thank you.